Hello, everyone. Good morning for one and good afternoon for others. Welcome to the first webinar of the MOOC Biobanking organized by the Institut Pasteur and broadcasted from Paris and also from Switzerland, Germany, and Spain. Today, we have four excellent panelists working on different scientific fields who will tell us the importance of biobanks for their research and their role during the health crisis situations, such as infectious diseases outbreaks that we have in the recent years. Our panelists are Olga Genilloud. She's the scientific director and head of the microbiology department at the Fundacion MEDINA in Granada, Spain. She has more than 25 years of experience in the discovery of novel small molecules derived from microbial natural products in the academy and in the pharma sector. We have also Amber Hallman-Scholz, deputy to the director at the German Collection of Microorganisms and Cell Cultures, DSMZ. Amber works on science policy to ensure the compliance with international legal frameworks that govern use and transfer of genetic resources, in particular the Convention on Biological Diversity and Nagoya Protocol. We have also Sebastien Gagneux, a professor at the Swiss Tropical and Public Health Institute in Basel, Switzerland. His research focused on the ecology and evolution of Mycobacterium tuberculosis. And last but not least, we have Christophe Batejat. He's the deputy director of the Environment and Infectious Risk Unit at the National Reference Center for Urgent Response to Biological Threats at the Institut Pasteur in Paris. His unit specializes in the identification of pathogens during emergencies. So we can start our first questions that were collected from the discussion forum of the biobanking course. I want just to point out that part of our audience participates in the syncytial virus and tuberculosis surveillance network of South Africa, Madagascar, and France, and others come from the University of Buenos Aires in Argentina and other countries in Latin America. So we have some questions for Sebastien that were made in the during the course of the MOOC Biobanking. And they are, what are the contributions of biobanks? What contributions can the biobanks make to the research of Mycobacterium tuberculosis and other pathogenic bacteria? For example, resistant to antibiotics, evolution, etc. And which is the importance of the establishment of international biobank networks in the fight against infectious diseases like tuberculosis. So you have the floor, Sebastien.
Thanks a lot, Liliana, for this introduction, and thank you all for joining this session. It's a great pleasure and an honor to be part of this. And so I received these questions, I think yesterday, and I was thinking about how best to answer. And then finally, I thought perhaps I answer the second one first, because this is not, specifically to tuberculosis. In fact, as we are experiencing this ongoing pandemic, maybe this is actually the best example of how biobanking actually can contribute to solving an important problem. Is that perhaps many people wouldn't think of biobanking when we start reading about the alpha, beta, gamma, and delta variants of the SARS-CoV-2 virus, but I think this is exactly what we're talking about. In other words, if you have a new pathogen emerging, and usually these are viruses minus, then it's actually a good idea to be able to collect these viruses and to look at them properly. And I think what's extremely unique about what we're experiencing today, I'm sure some of the colleagues here would agree, I think it's the first time that this is really happening in a real time. right? And that's really thanks to these new developments in the sequencing world. But not just that, it's also collaborations, right? So all around the world, people actually coming together, and they're not only collecting these viruses and sequencing them, but most importantly, which is essentially part of the biobanking exercise, but it's also about sharing the data, right? And it's only, I think, only by sharing data that you really can get the full picture. So I think that is an important point, that biobanking, maybe one of the key home messages, if you will, biobanking just per se, for the purpose of biobanking, is obviously not super useful if you're not going to use what you're actually banking. In other words, samples or specimens or strains, be it viruses, parasites, or bacteria. I think it's very important that you plan, that you know what you're going to do with this, and actually that you actually do something with it. And again, whether it is real time or not, that's a special situation. Ideally, if you have a pandemic, then it's going to be real time, and you can actually sequence these samples right away, and you can inform, I mean, scientists obviously, but it's all the way to politicians, right? So I think it's very interesting, very unique, actually, activities or almost experiment that we actually witnessing in real time, and which I think nicely represents the usefulness of some of these activities. How this relates to tuberculosis, because that's what I'm working on. There, it's real time is not so easy. We would like to be able to do that, right, with Mycobacterium tuberculosis. It's a bit more tricky than with viruses, just to get these bacteria collected in the first place is much more complicated. But nevertheless, it's being done. And I think it's a good idea that it's being done. And I would like now answer partially the second and the first question is with just giving you an example, which is very close to the work that we have been doing in our group, which really relied on biobanking in a sense. When I started to work on tuberculosis about 20 years ago, and the very wide dogma, literally, I mean, for most people working in the field, I mean, mainly basic scientists, microbiologists, but also immunologists, was that Mycobacterium tuberculosis essentially was a clone. In other words, that one strain, and it literally quotes to some famous microbiologist, I won't say the name here, that said, you know, if you have seen one M. tuberculosis strain, you have seen them all, right? And that was very much the view 20 years ago. And that has a whole lot of implications in terms of, be it, I mean, down to the development of diagnostics, drugs, and vaccines. You say, okay, if you just work with one, and usually it's actually a laboratory adapted strain. I mean, for the MTB aficionados here, H37RV, or maybe it's another one, Erdmann. And most of the biology that we know about this pathogen is based on these two strains, which are extremely lab adapted, haven't seen a human being for maybe 100 years. I'm not saying that it's not useful, but if you then think that you just can extrapolate everything that you know about what you learned about the laboratory adapted strain to the real world, you might be surprised to find that actually differences, right? And I think to go back to the biobanking thing, it's really thanks to efforts from many, many people all around the world who actually went through the hassle of collecting strains from TB patients and actually stored them. That we then gradually, we and others, and many other groups, of course, started to actually resuscitate these bacteria, which was stored sometimes also quite some time in the past. And of course, yes, sequence, analyze them in all kinds of different ways with phenotypic assays, etc. And it clearly emerged that it's actually not true that tuberculosis strains are all the same, right? But by now we fast forward 20 years later, I think now the consensus is that it's really important to take into account the natural variation that you actually see in M. tuberculosis strains. And to other bacteriologists, this might sound like, oh, I mean, it's kind of obvious. If you compare two E. coli strain, you're going to see huge differences, which is rather the rule rather than right, the exception. So TB is very, very similar, right? One strain compared to another. And there's a few other bacteria which are the same in the sense that they show very, very little variation compared to other bacteria. I mean, very little is relative. It's always the question of be compared to something else. But that's not to say that this variation is irrelevant, as used to be thought in the past. And if it weren't for this biobanking exercise by many, many groups, then people wouldn't have the resource actually to start to looking at these things, right? And so now we have multiple lineages, and we actually keep finding new variants out there. And that's only possible if you really have access to a large collection of many strains from all around the world. And that's actually key, right? If you just keep sampling the same area somewhere, I don't know, in Western Europe, you can keep finding more or less the same strain. That's not entirely true, because the immigrants, this is again something special by TB. Most TB cases in Europe are immigrants, so they will bring their own TB strains from somewhere else. But still, it's important to have global representation. I mean, that's another answer to some of the questions. Depending on the question that you have, right, make sure that your biobanking is comprehensive, in the sense, try to really capture the whole diversity which is out there. Don't bias your sampling just because it's convenient, because you're gonna miss important points. And I don't know, I'm not looking at my watch at all. So Liliane, you have to tell me when to stop.
It's okay. You're okay.
I'm just talking. So, and then, so that this is one example, for instance. If you really want to convince the world that TB is not a clone, then you need biobanking. I think that's what we managed to do. Now, there's other things. For instance, if you're interested in outbreaks, right? And again, so this is what we're seeing with COVID, right? And that's applicable to essentially all infectious disease. By definition, an infectious disease is infectious, therefore is being transmitted from one person to the next, right? That's universal. And if you want to understand who is infecting whom and why, and if you want to try to do something about it, then the only way to know this is by looking at the pathogens. And if you don't have the pathogens, then you might as well just guess. And that's the other reason I think biobanking is a good idea, that this would be perhaps on a more local scale, maybe in a particular city, on a region, a country. Depends on very much on the pathogen, right? If it's something which is like a pandemic, then you need a global approach. If you know it's some kind of food outbreak in a particular school or military camp or whatever it's going to be, or meningitis outbreak, right? Then you maybe focus on a particular local area to try to really understand what the basis is of this outbreak. So I think conceptually also it's important to keep in mind these, perhaps you can call these scales in terms of the sampling. You're interested in global questions as opposed to perhaps local or national scales. And I think all of these have implications of in terms of how you're going to organize your biobanking. And then has immediate, and I'm not the expert, I'm sure many here are obviously this has huge logistical consequences, making sure that the whole numbering system, your barcodes and whatever tool you're going to use needs to be adapted to these scales. And if it's a multinational or international exercise, then this is of course more complicated, because you have to ensure that the standardized ways of keeping track of your samples, etc. That's of course extremely important, right? To document everything. Particularly very old collections can be extremely useful, right? If people sometimes go back in time conceptually, you can think of these almost like fossils, right? You go back, microbes you can store for a very long time, and be it freezers or maybe also in the natural world. And sometimes there are questions. Actually, there's one point I would like to make. When I again started to work, and we're all excited about sequencing, right? In the early days of sequencing, of course, sequencing was so expensive that it was actually the sequencing which was the limiting factor. And biobanks already existed, right? There were big collections of bacteria and viruses all around the world, but there was no way that you can actually sequence all of these because sequencing was too expensive. And argue that today it's the other way around. Today the sequencing capacity is so huge that in fact it's rather the strains or the collections which are limiting. If particularly the ones which were properly collected and not biased, and et cetera. So that's why it makes the biobanking even more precious, because now the technology is not the issue anymore. Now it's back to the biological question rather than the actual sample, the material which becomes limiting. I think that's quite an interesting development that science has been going through, particularly as far as microbiology is concerned. Right, I think that's about what I wanted to say.
Okay, thank you, Sebastien. It was a very nice input and enlightening response of the role of biobanking in the research of tuberculosis. So thank you very much. Now I want to go to pass to Christophe for the questions made by our students, which includes which is the importance of the establishment of international biobanks network in the case for the fight for against viral diseases in all the viral outbreaks that had happened in the recent years, and what has been, for example, the specific case of the viruses biobanks during the COVID pandemic, especially the project EVA-G, which is the European Virus Archive, which is a European project for virus biobanking, and which was the role of these kind of projects, especially during the first stages of the pandemic. So you have the floor, Christophe.
Thank you. Thank you for inviting me. About the importance of biobanking, everything has been said. It's about sharing. Sharing biological materials, viral strains, but although diagnostic reagents, and sometimes in the case of European Virus Archive, you can share trainings too. Sometimes you can share more than just biological materials or training. You can share experience and discuss with other scientists. This is very important in peacetime, but although in pandemic time. That's what we have seen during this pandemic. A lot of things have been shared, especially through the European Virus Archive. I don't know if you already talked about it, and if everybody knows how it works.
No, not yet. You can maybe develop a little bit on this.
Okay, so the European Virus Archive is a kind of virtual biobanking structure. That means that every participating lab or institution keeps in his fridge, in his -80, his own strains, and the website is the virtual biobank. And when sharing these, every lab keep his own samples and strains, and will send them to the client. So this is a European project, and it has been working for years now. And during the different pandemics or epidemics, it has proved his interest. And for example, with Zika, a lot of things have been shared this way. And for the COVID pandemic, many strains and diagnostic reagents have been shared this way too. So sharing of your strain is very important for diagnostic development and validations. For every new variant or for every new strain, you have to check that your diagnostic techniques are still working. For the development of vaccine, antivirals, and a lot of research work, you will have to use the different viral strains circulating in the world. So many things are going through biobanks. Our diagnostic reagents have been shared and sent all over the world through biobanks and European Virus Archive in particular. And external quality assessment, although I think this whole use of the biobank were key to the development of the response in the early stages of this pandemic, and still now. I guess it's all.
Okay, Christophe, thank you very much for your inputs.
Welcome.
Very useful for our students to know the real role of the biobanking in the pandemics, and not only COVID, but also in the previous one related to Zika and perhaps Ebola, which had place in some years ago. So now we are going to Olga. Welcome, Olga. We have some questions from our forum of discussion of the course, in which they ask, since biobanks are considered a source of biological resources, services, and data, it's not only the let's say the microorganisms which are the valuable asset of a biobank, but also sequences, the techniques for conservation, and the taxonomy information about them. Which kind of specific research that is carried out within the biobanks? Like, for example, the microbial natural products discovery of the microbiota studies could become a source of sustainability for the biobank. And what has been your experience on that aspect?
Thanks so much for the question. Yes, in fact, we are now entering in an aspect of biobanks that is completely different in the sense of how we can exploit the microbial resources. And this is becoming more and more evident that they have a major role to play in the field of biotechnology, in the discovery of many high value products. Of course, biobanks, from the perspective of taxonomy, of reservoirs, of strains of reference, have been key in general microbiology, just to have these reference materials from them. But also, more and more they are becoming, and also what has been already mentioned in the sense of more tools are available, more information from the genome of all these strains are now becoming can be accessed from the biobanks, and there are central pillars in the activity that is being performed today in this field. From my perspective, I've been just experiencing how to work in the drug discovery from microbial natural products, as it was done in the past, also in the pharma scenarios, but also today this activity is being performed by many research teams, and they have referenced the biobanks to provide these materials they need to constantly just check in their research. So considering that these biobanks have been collecting for decades key reference materials in this field. And now that there are many and more and more projects that have been engaged into getting access to the whole genome sequence to get much more information about all the not only the primary metabolism, but also all the secondary metabolism of all these strains. And we have the tools to mine into that. This knowledge that's been continuously growing has also been evolving with the trends in how new assets are being discovered. So the way biobanks are taking a role in this field today has been evolving with the evolution of the technologies that are now applied, and they are the starting point of many different type of projects just to go deeper, as they are unique reservoirs of diversity. From the perspective of natural products research, of course, anything that we are doing is focused on trying to identify novel chemical diversity when you are interested in the small molecules that are produced by these strains. So this is why that the current scenarios are very much different from what we were doing several decades ago, when the strains were tested without really knowing too much about their not only just their position or their taxonomy, but also in terms of their physiology. And a lot of resources were invested in going through the metabolites that were produced by these strains. Today, the approach is much more different. I mean, is much more centered on the individual strains, selecting the best candidates, because we have the tools to mine into what they are potentially able to do. And then from there, just to use all the technologies we have at hand to try to make them express diversity of, this can be small molecules, this can be enzymes, a lot of applications in many different type of fields. This can be human health and animal health, in agriculture, in food preservation, cosmetics. I mean, there is a long list of applications from the different products that can be obtained from microbial sources, and that's where the biobanks can have a role to play. They can develop, of course, also a business on that, in the sense that at the end you have products that can be just developed for a potential use by the society. And so that's one of the strengths today. Of course, you need to align not only the capacity to manipulate and also to grow and to learn more about all these strains, but to try to align this with other expertise that sometimes is not available in the research centers. But today, this is something that can be integrated with the different type of disciplines that are needed to perform discovery programs, just based on the interest in looking for new molecules coming from these assets. So I see that, in fact, from our own experience, and this is something that we are doing ourselves in our center. We are exploiting one of the largest microbial collections. Of course, this was not built as one reference biobank when you have been very well characterized in each individual strain. But in this case, the approaches that we have been developing could be for sure been translated to any biobank willing to exploit the resources they have as in their deposits, and this is something that is extremely useful for anyone working in the biotechnology field, having a place to come back, having reference material that can be used in any type of research.
Thank you, Olga. I would like also to do another question about this kind of research for biobanks, because most of or many biobanks that are currently, let's say, working on research, they are funded by public, let's say, funds, either the government or multi-governmental funds like the European Union, or maybe locals or regional funds. So this kind of research, how is let's say, how can we valorize these or exploit economically this kind of research, and also in terms of intellectual property, when there is an association of public organization with privates, which are also supporting this kind of research, which is, in your experience, how these situations can be handled?
Yeah, these situations are very frequent, just to be found in any consortia that we are just developing research with institutes and also with companies, even working from Horizon Europe will be the same. So our experience is that, in fact, when you are just performing research within this consortia, you normally agree what you're going to be doing with these assets that are coming out from the project, not only just the strains themselves, but also any small molecule or any enzyme and the type of application, and which are going to be the rights of any of the end users. So that's something that need normally to be agreed by the consortia. In other cases, when the research has been performed by the research teams alone, and then you have the companies willing to get access to these microbial strains or the small molecules that have been showing any potential activity of interest as a starting point for their research. Of course, the IP and the IP rights remain on the team that has been discovering these molecules, have been isolating these strains, and they have the capacity to license a use to the industrial users. On the terms, they will need to agree. Of course, the terms will be varying very much depending on the sector of activity. It's completely different to deal with the pharma sector or the agroveg or the cosmetics. Each one is defined according to the market. But this is very straightforward, the path to follow, in the sense of being able to transfer the knowledge that has been just generated with the public funds, and managing to have these assets become available to or just to public-private partnerships, or just to the private sector, because you can define very clearly the terms about how this is going to be used, in which type of research, what can be the future returns if this product is managing to get to the clinic, to get to the market. And at the end, you can negotiate from the beginning what potentially can be these returns for the research organization. So this is also a potential source of benefits, and not only just economical benefits, but also for the potential end users being able to have from a research that has been originated from a public funding, not staying just blocked in one lab. There are all the tools to manage to develop this into new products, if there is an industrial partner that is interested in exploiting that. So the tools are there. We are using them on a routine basis. In the case of our institute, is a private foundation. We are non-for-profit. We are working, almost half of our research is being performed with companies worldwide. And these are the normal agreements that we establish, because at the end we are not going to reach ourselves the clinic. So you always need to transfer your IP. You need to negotiate what are going to be the terms to do that. So this is the best way to work together and to manage to get that any potential product of high value manages to get being developed by those that can do it, because normally in the early stages we don't have the resources to do that.
Thank you, Olga. Thank you very much for your contribution, and also for telling us how is the contribution of the biobanks, and how is the way to valorize our research results and putting them out for the industry, or in a way to increase as well the profit of our research and resources in the biobanks. So now we are going to go to Amber. So Amber, welcome. We have several questions regarding to Nagoya Protocol, and also access and benefit sharing in our course. But basically, this is a subject that is not very well known by the research in general. So unless in some parts of the world that they are not very used to these kind of things. So basically, I would like you to just just to say how is the importance of the compliance of these kind of legislations, and also how is the importance, or not importance, but how is been the effect of a situation of health crisis in like the one we are living now on these kind of legislations, how it's been affected, even the legislation to the pandemic, or the pandemic to the way we fulfill these kind of requirements. So you have the floor, Amber, and thank you.
Perfect. Thank you, Liliana, for the kind invitation to join you all. I work a bit in a niche field here in the Convention on Biological Diversity and the Nagoya Protocol and access and benefit sharing, as Liliana explained. So if you'll be patient with me, I'm going to give you a little bit of background on the Nagoya Protocol and access and benefit sharing to be able to come back and answer the questions around benefit sharing from pathogens, from SARS, from Ebola, or SARS-CoV-2, the coronavirus, and other pathogens. So I thought it might be helpful to give you a little bit of context first and foremost from our collection here at the DSMZ here in Northern Germany. So we're a pretty big microbial collection. We have about 10,000 customers in 86 countries, about 2/3 international and about 2/3 academic. So we really have to figure out what the international legal landscape is. And one of the most important things we have to do is to make sure that we're able to offer the scientific community products that they need, microbial resources that they need, and that are compatible with the scientific system and with the legal system. And you might have already covered this in your other lectures, so my apologies if you have, but just as a first note, why do scientists put microbes in a collection? The most important reason is, in the case of microbiology, because of the International Code of Nomenclature on Prokaryotes, the ICNP. And that is a code that tells us scientists how to validly publish a new species and identify new biodiversity. So the rules go, you have to, number one, deposit that new species in 2 internationally recognized collections. And recognized means recognized by the World Federation for Culture Collections, that's the WFCC. Perhaps you work at collections that are WFCC members. Number 2, those 2 collections validate the depositor's new species by giving the depositor a certificate of deposit from the collection. So it's a DSMZ letter and it says, yes, we have received this bacterial species for deposit, and it is valid in terms of the ICNP rules. And number three, that strain has to be available to the scientific community without restrictions. And here is the touch point with the Nagoya Protocol rules, because this phrase makes it very difficult for both the scientific laws or policies to be compatible with some of the international laws. Also, just a short note in case you haven't already discussed it, plant, animal, and fungi taxonomy follow very similar systems, and other reasons that people deposit them in that our main argument for why our collection gets the public funding that Liliana talked about is because we know it enables the scientific community to be able to function in a way that is reproducible. Because without these standardized high quality materials, we know that an average of $10 billion is lost in the entire global marketplace due to using incorrect biological reagents and reference materials. So if you're looking for arguments for your funders of why you need to exist, this idea of scientific reproducibility and standardization is a really important one. So now I want to jump into the Nagoya Protocol. This is an international supplement to the Convention on Biological Diversity, which is an international treaty under the United Nations. And it has basically two aims: to provide legal certainty for the actors that are involved in the use and international exchange of genetic resources, as well as traditional knowledge. Although for microbes, we most often focus on genetic resources. It tends to be that for plants, traditional knowledge is a more relevant topic. And also then to specify the means for the sharing of benefits. So what Olga talked about in terms of the ability to license products and to be able to say, I as a collection or I as a user own this, actually the Nagoya Protocol goes a step above that and says, well, actually the country from which the material comes from has the sovereign rights and the opportunity to claim benefit sharing for itself from anything that comes out of that biological resource, meaning commercialization, but also for non-commercial research. What is very important here when you look at the definitions is you do not see that pathogens, for example, are excluded. Everything that's alive, except for humans, not human DNA, not human genetic resources, but everything else is covered under the Nagoya Protocol, and also independent of whether or not the use is non-commercial or commercial. Everything's underneath that. So what we have to understand a little bit is how then the Nagoya Protocol is supposed to work in practice. So the first thing you have to figure out is, are you working in a country where there is national legislation in place? Most of the time you'll go to a place called the ABS Clearinghouse. You can type that in Google right now and see what that is, and that will give you some first level information about whether or not you have it. Liliana sent me some of the countries that are represented here today: Madagascar and Argentina, Brazil perhaps, and those countries do all have Nagoya regulations. So if you're working in a country there, you need to go to the ABS Clearinghouse and figure out what your national legislations are. And then the Ministry of Environment in most cases is responsible for issuing different types of documents called PIC or MATs, which stand for prior informed consent or mutually agreed terms. Those can be all tucked into one single document, or it can be multiple documents. And the scientists would go out and do their sampling, and then the research and development can legally take place. So one of the things that the DSMZ has done in the last few years is to become a registered collection under the EU law called 511/2014, and that is the European Union's implementation of the Nagoya Protocol. And what that means is that we have voluntarily tried to figure out this mess for the users. So we've taken care of this aspect before we accept deposits into our collection. And the reason that that's important is under the EU law and under many laws, a biobank or a culture collection is actually not obligated to follow the Nagoya Protocol because they're holding stuff. They're simply a collection, sort of a static, if you will, and that is not the act of utilization, not the act of conducting research, which means they're not obligated to go find those Nagoya documents, that PIC and that MAT that I mentioned, which means the user actually that has something from a biobank needs to go back to the original depositor and ask, how do I comply with the Nagoya Protocol? This makes it very difficult. And so what the registered collection law does under the European law is to say, okay, the collection itself takes care of attaching the documents for ABS, for the Nagoya Protocol, and then passes those on to the end user, which makes the end user's job of being compliant much more simple. So that's what we've done here. And just to give you an idea of how we implement that in a flowchart, we have a microbial resource coming into our collection. We ask whether it was collected before October 2014, that's the date of entry into force for the Nagoya Protocol, and if that country was a party to the Nagoya Protocol. If it was, whether when the sampling, when the collection of that microbial resource happened was also before or after the date of entry into force of that. And if it was, then we ask whether or not that country has given free access, has no ABS legislation. So that's true, for example, here in Germany, we do not require PIC or MAT documents like other countries do. And so if free access is given, we don't have to ask for any more documents. But if access is regulated, so let's take the example of Madagascar or Argentina, then we would now need to ask our depositor, can you please show me your permits, your ABS permits, these documents that are required under the Nagoya Protocol? And then we check those documents, and then we either accept it into the collection or reject the deposit. I'm going to skip that one. So now I'm coming back to Liliana's questions about pathogens and whether or not they are in scope of the Nagoya Protocol. As I mentioned earlier, they are. And there are groups out there that really watch out for this. So we know, for example, in the case of Ebola, there was a relatively high profile newspaper article about Ebola samples being taken out of Africa. And this quote I find very interesting about, I don't know if it's this patient in the picture, but a patient does not know that the Ebola virus isolated from her blood is advertised on the website of the European Virus Archive for sale by Germany's Bernhard Nock Institute at a price of over 3,000 euros for 0.5 milliliters, 170 times the price of gold, right? So there are institutes or groups out there that are very much concerned about the legal acquisition of samples in general, but in particular for high profile virus outbreaks. And then there are papers like this that come out where it says, you know, there are companies, in this case, this is an American company called Regeneron Pharmaceuticals that was avoiding the benefit sharing back to, I think in this case, it was Nigeria or perhaps New Guinea, where there should have been benefit sharing back to that country because these samples were collected by someone that should have gone through the Nagoya process. And so now if we look at the coronavirus, and as Christophe and Liliana mentioned, there are a number of virus products available in the European Virus Archive. So we've taken an inventory of all of them and we've asked this question, and my apologies for the small size of the text, you can see we have a lot of products. Actually, the countries where these have been collected in so far, you can see here perhaps France, Slovakia, Germany, the Netherlands, Italy, France, the United Kingdom. These countries do not require access and benefit sharing. And so the interesting thing about pathogens, especially highly infectious ones, is that it is much easier to get them from patients that come and land in your country than it is necessarily to go back to the original country of origin. So even, for example, when we have new variants coming out, like in South Africa or in India, where countries that would require access and benefit sharing permits, because those people, somebody from that country gets on a plane and lands here in Germany, it's actually very simple to collect here in country. And therefore, the way that the law is created, it is not then required to have the access and benefit sharing documents, which actually means that the countries that don't require ABS are perversely incentivized to do the collecting, even though all of this is a global sharing thing. Liliana also asked me about the rules underneath the Nagoya Protocol that are supposed to make access in case of public health emergencies more possible. And indeed, there's an article in the Nagoya Protocol called Article 8, Special Considerations, in Article 8b, that in the case of present or imminent emergencies that threaten or damage human, animal, or plant health, that parties should make special considerations. But the reality is, very, very, very few countries have done this yet. And so then when you land in a public health emergency, like in the coronavirus or Ebola or Zika, the countries do not yet, at least, have special permissions in place. And so these pathogens fall under the exact same rules as any other kinds of biodiversity in that country's border. And finally, I wanted to just point out that, again, to the earlier points made by panelists, this is a global community. So you can see here on this map, the products that have been shared with many countries around the world from the European Virus Archive that have either been for free or where the person had the option to receive it for free and then chose to actually pay for various licensing purposes. And so this is truly a widespread role of a biobank in this case, where the legal framework unfortunately doesn't quite match up with the use. I hope that was what you were looking for, Liliana, but I'm happy to talk more if I can help.
Thank you, Amber, for this very nice presentation, and also for showing us or showing all the students which is this legislation about. I have one question, because you said that this is a legislation that is from the United Nations. So of course, some countries choose to not follow it because they have their own legislation to regulate these kind of processes. But in case they choose to follow, you said that is different, that the law and the way that the countries implement the law. So in some countries, the implementation of this law has given some kind of problems, especially for the development of research. It's more complicated to do research. Do you have anything to say about it?
Yeah, the interesting thing about these international treaties, the way that they're drafted, is they give parties options to which the individual country can decide. So the Article 8 that I just showed you is an example. It recommends very strongly that, for example, for research being done for biodiversity and conservation purposes, that countries should consider these exceptions. And the same thing again for public health. But if the country, when they write their national legislation, does not do that, then they chose not to do that. And that's under their sovereign rights to choose how they want to implement that. And so then as scientists, we're left in this position of saying, you know, at the international level, it was strongly recommended that you make exceptions for public health. We don't have any in our country. Can you please address this important need? And if they say no, well, you only have the same, tools available that any normal citizen has, which is to say, I want to elect new leaders, I want to be involved politically, but ultimately that's where the decision lies. And our hands are a bit tied as scientists when we're caught between the laws of our country and the international standards of practice for exchanging organisms and for international cooperation. But I think what's perhaps the most important thing I'd like you to know about this area is that if you're working with biological materials, even if you don't send them outside of your country, it is very important that you go to the ABS clearinghouse and figure out if your country has laws around ABS, and that you learn what those laws are supposed to do. Even if they're cumbersome, even if they're hard to implement, that's our job, and it can get you into a lot of trouble if you're not yet aware of it.
Okay, thank you very much, Amber, for your answers. I think we can maybe just make one question regarding this, because it's also very, it was, let's say, it was very often in our forum, just to know if the situation of COVID, the pandemic, affected the way the material or the samples that you use for your research was in any way affected, because either was facilitated by, in the case of viruses, because it was a priority for the research on COVID, or in case of other organisms or pathogens that were not priority, were not actually easy, or were even impair the transfer of this material, or how your research was carried out during this last year of pandemic. I think it's a question for maybe Sebastien and Christophe and Olga as well, in case your research was also affected by the pandemic. Let's start with Sebastien.
So whether the research, yes, I think it clearly was. I mean, particularly because some of the work we do with partners in Tanzania, I don't know if some of you followed what happened in Tanzania. It was a bit of a special case. So just to summarize a bit, I mean, there the former president actually declared that coronavirus didn't exist, and that's sometime in April, I think. And then people stopped testing. But of course, behind the scene, people still were afraid. And of course, our scientists, colleagues, had a bit of a hard time, because they knew that on the one hand, there was this virus circulating. On the other hand, from a political point of view, they were not supposed to talk about. And then, as you might have heard, then this president actually died. And officially not from COVID, but unofficially, yes, from COVID, and etc. But it's a bit of an anecdote, but yes, I think everyone who was doing so-called, sometimes we call it field work, whatever that means, right? Particularly in areas where, for instance, tuberculosis, but I think the same is true for malaria, people working in malaria or the HIV field. I mean, we know all of these because of the issues of health systems, right? So arguably more important than research work, the health work was disrupted, right, because of COVID-19. So many diagnostics places didn't continue. For instance, stopped diagnosing tuberculosis patients because they were focusing on COVID. Obviously, that was not the case in Tanzania, because officially there was no COVID. But for instance, in India, we know, you might have seen these figures, right? The diagnosed number of TB cases diagnosed during 2020 dropped by a third or so, which obviously is not because there's suddenly no TB anymore, just because TB patients are not diagnosed. And then indirectly also this would affect the work. I mean, we don't work in India, but for instance, we also work in Georgia, not Atlanta, Georgia, the former Soviet Republic, country of Georgia. And they also was not so heavily affected by COVID at the time, but still it had a direct impact on the work that we're doing there with our colleagues.
Okay, thank you, Sebastien. Olga, do you have this experience in your research? I cannot hear you, sorry. Your mic is off.
Sorry about that.
Yeah.
Yeah, I was explaining that in our case, we are not working directly with samples that have been directly affected by all these restrictions. In our case, it has been most just by the impact on the administration of granting permits, in transferring samples from one country to another. I think we have been affected as anyone working in this field, because the processes have been much slower. And in this case, well, at the end, everything is being impacted in a certain way. But one point I would like to also take from what Amber has been explaining, also just connected with the Nagoya Protocol and what I was mentioning before, if you permit me to divert a little bit from the question, is of course also the importance for the exploitation of any of these resources, just to be compliant with all the Nagoya Protocol, and not only just the Nagoya Protocol, but also the CBD that previously was established in the 90s. So having this traceability of all the microbial resources in any biobank is going to be essential, just to avoid any potential future conflict, because of course there will be no permit allowed to commercialization, to registration of any product if you cannot guarantee that you have been getting access to these resources on a legal basis. So this is something that is essential. It's essential for anything coming after the Nagoya Protocol, but also from all industrial sectors. Having a good trustability of all your strains that you are working with is also essential. And at this case, we have been in our case from our large collection, we have been undergoing an audit just to double check all the legal status of each individual asset in our collection, just to be sure that at the end you are not putting in risk any of the research and the investments you are doing in the future. So I think I would like just to mention that because it's essential. And as a resource, we are transferring the obligations to any user that will be developing a product coming from these microbial sources. So is a key, a central point on anything that we are doing, just to be compliant with that.
Okay, thank you, Gloria. Olga, thank you very much for your answer and for complementing as well what Amber said and applied to your case. Christophe, you have any very quick experience about the sample transfer in your case? It was facilitated or impaired during the pandemic?
Yes, most of our transfer of samples have been on COVID-19. So most of the time it was facilitated by the authorities. So that was a good point. During another pandemic, which was Ebola, everything was more complicated because of the transportation companies who didn't want to transport Ebola samples. So depending on the pandemic, of the epidemic, you can have difficulties and sometimes unexpected difficulties with the exchange of samples.
Okay. Thank you very much. I think we are already 2 minutes after the end of our seminar. I would like to thank all our panelists. Thank you very much for your inputs, for your contribution to this discussion. Thank you for answering the question to our students. And thank you for your time. I would like to thank also to Odile Cismeiro and Leonardo Alvarez, which are part of the technical team behind all this seminar. Also to Monica Sala and Thierry Lang from the Pasteur Teaching Direction. To the teaching team of the MOOC Biobanking, Raquel Hurtado, Mary Piña, and Marie-Noël Ngoyor. To the Dominique Clermont, Muriel Guguer, and all the SERVIP team. And also to the two wonderful community managers, which are with the students in the forum in the MOOC Biobanking, which are Ilse Lowe from South Africa and Caroline Entrecasi from Argentina. And I wish you all the students of the MOOC the best of luck during the last weeks of the course. And I wish you all a very nice afternoon and see you next time. Goodbye to everybody.